On the other hand, we include either of these two blockers of UPS, we now get the protein back. So this is all consistent with the mechanism we envisioned, that in the absence of epitope, the protein is being degraded through the UPS system. Okay, now looking at the sequences, these mutations that then gave us the conditional stability, we were happy to see that these mutations were in the framework, or the heavily conserved regions of these camelid nanobodies. So these are three different species of nanobodies, and what we're showing here is the framework regions, which are the heavily conserved regions among all nanobodies, and this is where these mutations map. So these are the most important of the mutations we found. So this was great, because what it told us is we might be able to substitute these mutations in other nanobodies without having to do the laborious screening procedure. We could just take any nanobody whose sequence was published, swap in these mutations, and see if we now had conditional stability, and we could test that by adding the epitope. That worked quite well. The majority of nanobodies we tested were conditionally stabilized by these mutations. And I'll show you some of the applications then that we put this to. So I'd like to just show you this set of panels. This was now to make a flip recombinase dependent on GFP. We use a destabilized GFP nanobody. And look at the activity over here. This is again a readout for luciferase after flip activity. We took flip and we fused it to either a single destabilized nanobody or a double. And we asked, is there GFP dependence or recombination? And as you see over here, we have a lot more recombination when GFP was present versus when it was absent. So this is about a 600-fold induction of flip activity by having this dual nanobody domain, and again having these destabilized domains. If we don't use the destabilized domain, here's the normal nanobody. It doesn't have GFP dependence for activity. Now this is great because first of all it gave us this reagent, FlipDog, which again you can all use in the same way you use CreDog. You only have to deliver one protein. It's active. It can be used again in a combination with, let's say, a Cre strain. If you have a Cre plus a Flip mouse cross and you introduce this, you'll have again a Cre plus Flip intersection for your genetic manipulation. The second thing this showed us though is that we could put two nanobody domains on a single protein, and that gave us the possibility of doing an AND function, again to create more specificity in the Flip readout. So we had nanobodies for a number of different proteins. I'll just show you one example over here. We had a nanobody from the literature for an HID capsid protein. We introduced the conserved mutations, and we found antigen dependence. We then took that destabilized anti-HID domain and fused it in tandem with our anti-GFP destabilized domain, again fused to flip. And we asked, is flip activity stimulated only when both antigens are present? And indeed that worked. That's this column over here. Again, these are in vitro readouts of luciferase. If both antigens are present, we have a great deal of induction. If none of the antigens are present, we have almost no activity. If one is present, we have a little bit of activity. So this then opens up the possibility again to gain access to a more specific subset of cells by using a combination of epitopes to turn on a dual nanobody fusion protein. So this is the last application I'll show you then. This is now to use these single nanobody fusions that are dependent upon the presence of epitope, and to use them as detectors. Now this is useful in a number of applications. If you just want to know if your antigen is present in a cell, let's say you're working in a system where you're looking at induction of endogenous protein, one of these introduced intracellularly will give you the ability to see the turn on of your protein. This works much better than just putting a fusion protein in, because if you put a fusion protein in that was not destabilized, so let's say you had a nanobody to antigen X fused to GFP or GFP, that nanobody is stable in the absence of epitope, so your cell will have sort of a blue haze as a background. Here, if you put in a destabilized nanobody, there's no background. The nanobody goes away unless the epitope is present. So this is a very sensitive detector system. We wanted to test this on a virally infected set of cells. We went for an HID. We worked with Jonathan Lee and his postdoc Azad Hamad. And what we wanted to know is whether HID capsid protein, when induced, would enable cells to turn red if we put in our fusion protein, our destabilized fusion protein, to detect that. So we took a set of cells that were the parental line. These are uninfected, so there should be no HID epitope, and therefore no red from the fusion protein. That parental cell line is infected with HID to create a derivative line. HID in this scenario is latent, so you probably know that HID, once it infects cells, can go quiet, not express any of its genes. But for reasons that are unknown in a person that's chronically infected, they will reactivate the genome, and that's the point of study. People who are studying HID biology would like to know what activates. And this gives them a way, if they can study this with our fusion proteins, this gives them a way to screen for activation in a live cell population. You don't have to permeabilize the cells or do any of the other things that you have to do normally to see an epitope turn on, especially if it's an intracellular epitope like that HID capsid protein. So this cell line has a quiescent genome, but that genome can be activated by a chemical called DNA. So we took the parental cell line, the non-reactivated cells, and the reactivated cells. We introduced our destabilized anti-capsid nanobody, tagged in this case with RFC. This is just a transfection-controlled GFP. It doesn't have anything to do with the nanobody. We then introduced these constructs and then FAST to ask, would we get some red cells if we reactivated the HID genome? And you can see from the FAST plot, this is the area where the red protein was seen among the GFP transfected cells. The parental cells or the non-reactivated cells did not have any red cells. That's quantified over here. This is the reactivated HID-infected cells. This is the parental line. This is the non-reactivated infected cells, and again, the parental cells. So this is a very sensitive way then to test reactivation of HID, and it can be used in high-throughput screens. It's similar to be used to detect viral antigen in other infection scenarios. It also can be used, again, to look at the induction of an endogenous protein, if that's your interest. And this is the last experiment then. We wanted to know if we could induce Cas9. I'm sure most of you know about CRISPR. One can then get genome editing specifically in the cell that expresses the epitope. We again used our HID destabilized anti-HID antibody in a double fusion scenario with Cas9, the enzyme. We tested this in a tissue culture system where LAC-Z would be turned on the guide RNA was then directing the Cas9 to excise a transcriptional stop. So Cas9 activity is indicated by LAC-Z activity. We looked at a wild-type normal allele of Cas9 in this assay, and it worked quite well. So this is now LAC-Z readout. So this is the normal, non-modified Cas9 in the presence of guide RNA. We set that activity to 100%. In the absence of guide RNA, there's no activity. And this is our destabilized fusion protein over here in the presence of HID capsid. You can see we got quite a bit of activation, about 70% of what we see in the normal wild-type Cas9 allele, and it's dependent upon epitope. If we leave out HID, we get very little activity. So now one can then use these fusions to direct the genome editing. I think this could even be used in a scenario where one is trying to affect, let's say, deletion of a dominant negative allele for human therapy. And I think this